Now we're going to launch into the third and final section of animal physiology, and we're going to be talking in this unit primarily about the regulation of the internal environment of an animal, or what we also call homeostasis. So there's several different things that we can be regulating inside the body of an organism, and the primary ones that we generally talk about are things like energy. Now, this is something that we already talked about, and we're not going to talk a lot about, but most of the energy homeostasis we already covered in the metabolic section, so we're not going to come back to that very much here. But we also do talk about temperature regulation. Maintaining a constant temperature in an organism is really important because most of the enzymes that are operating in an organism's body are tuned to operate at a very specific range of temperatures. And so there needs to be some sort of temperature regulation. Whether or not that's physiological or behavioral can vary from animal to animal. And we will go on and start talking about temperature regulation later on in this unit, but for right now we're going to talk about something else, which is the regulation of osmolarity in an organism. And again, this is really important as well, because once again, living organisms are really dependent on water, but also not just water, but the solutes that are in the water as well. All the ions that we have in there, everything else is dissolved in the water, and a subset of these solutes I'm including here is pH, because really the pH is going to be arising from the specific kinds of solutes that we have dissolved in the water in the body. So this is part of that, kind of lumped into that osmolarity regulation as well. So let's kind of think about the different strategies, kind of from a bird's eye view, or kind of a global view, the different strategies that an organism can take to regulating the osmolarity of its body. So what we have here is just kind of a simple cartoon graph, where on here in the x-axis we have the osmolarity of the environment, and we can see that increasing to the right, and then looking at how the osmolarity of the body of an animal, the y-axis, increasing upwards there. And so we can have several different strategies. For instance, we can have what's known as an osmoconformer that will allow the osmolarity of its body to adjust to and stay isoosmotic with the osmolarity of the environment. And so one that does this completely, there's no regulation at all, we call this a strict osmoconformer. And really, in reality, nothing is a completely strict osmoconformer. It's a gradient, and so there's also what we call osmoregulators, that whatever you change osmolarity of the environment to, they're going to maintain a constant osmolarity of their body. So they're going to try to maintain a constant osmolarity, whatever that is in their environment of their body, despite how this might be changing in the environment. And if this is completely unchanged, we would call this a strict osmoregulator. Now again, just like a strict osmoconformer, there's not really anything that is completely strict at this. There's some gradation between these two different extremes in strategies, and really most things fall into some sort of limited osmoregulator here, where what we see here is an area in which it very well maintains or very well regulates its osmolarity, but outside that range, it loses that ability to regulate its osmolarity, and it will begin to vary with the environment rather than being regulated within some sort of certain bounds. And what these are known as is the critical osmolarity. Where this break point is. This is the osmolarity at which we can no longer maintain a constant osmolarity in an organism. So most organisms fit into this group where within a certain bounds, they're just fine. They'll go ahead and osmoregulate, but you take them outside of those bounds and that breaks apart. Okay, so to talk about this, we need to kind of set down, where are we doing this? Where are we trying to maintain this water and solute balance in organisms, and so we need to start talking about these different fluid compartments and exchange of water and exchange of solids between these different compartments. So for the moment, because there's a lot of different compartments and a lot of different animals, I'm going to stick to mammals for the moment so we can just kind of think about that. Now these are going to be very similar between different organisms, but there is going to be some change as well. So in mammals, we have the intracellular fluid. So this is really the cytosol and everything that's inside a cell. So that we can maintain fairly tightly. We also have the extracellular fluid. So this is everything that is also outside the body as well, or not outside the body, but outside the cells in the intercellular spaces. This is also called interstitial fluid. So interstitial fluid, extracellular fluid, this is everything that is outside of cells, but still making up the body and still making up tissues as well. Then we also have something called intravascular fluid. And so we're just going to make this a blanket term for things that are being circulated through the body in vessels. So in mammals, predominantly this is blood and lymph from the lymphatic system. These are going to be regulated in a slightly different way than the extracellular fluids or the intracellular fluids are going to be regulated, but this is the primary place at which we can do regulation. Because these intravascular fluids are being pumped and transported around the body, we can change them in one specialized location in the body, say a specialized organ for maintaining correct osmolarity. And then once we've corrected the osmolarity in these intravascular fluids, push them out to the body to places where they can then exchange fluids with the extracellular fluids and help maintain osmotic balance there, which then in turn exchange with the intracellular fluids and maintain osmotic balance there. So really this intravascular fluid is going to be a primary place of osmoregulation in mammals. Also, we have intraorganismal fluid. So this is fluid that is contained within the animal. However, it's not really inside any tissues, and it's not something that is going to be contained in something that we call tissue. And so, for instance, this would be the fluid that's in your digestive tract. So if you drink water, or as the bolus of food moves through your body, this is intraorganismal fluid. Also, we have fluid that's inside your urine that is being held in a urinary bladder. It's also intraorganismal fluid. Now, really, this is kind of an intermediate between fluid that is actually inside your tissues and fluid that is in the environment. And so we're going to be concerned with this, and this is going to be the byproduct of osmoregulation. But it's not going to be something that we are trying to really tightly regulate, because it, in some ways, is not really a part of the body at this point. And so the regulation on this intraorganismal fluid is going to be a lot less than what we're seeing here in the intracellular, extracellular, and intravascular fluid. There's a lot less regulation here. And in fact, this intraorganismal fluid will often be varied in its composition a lot due to the regulation that we're doing in the intracellular, extracellular, and intravascular fluid. Essentially, the waste products that we are generating by trying to regulate these other three compartments are going to be put into this intraorganismal fluid and stored there. Then we also have extraorganismal fluid. And this is the fluid that is outside the organism, that is interacting with, and is going to eventually form the environment in which this animal is, and that it can do some exchange with in terms to adjust the osmolarity inside the fluid. So this is really obvious in aquatic animals, because that is the water that they're in. However, this is a lot less intuitive when we start talking about terrestrial animals, but there's also extraorganismal fluid that's important there. So this is going to be the fluids that will sometimes cover them as they just live, like rain, snow, other things like that they interact with. That's to a less degree than fluids that then they'll also drink. So for you, Mountain Dew might be an extraorganismal fluid. It's outside. You can do some exchange with it as you interact with it, but it's not inside your body as of yet. And so this is really part of the environment, not part of you quite yet. And it'll do most of its interacting with you, at least for terrestrial animals, if it becomes eventually this intraorganismal fluid. Okay, so that's the basic compartments that we're talking about. We talk about fluids that are going to be undergoing some sort of a role in osmoregulation. Now, there's different ways that we can be exchanging fluids between the extraorganismal fluid and what's happening on the inside of you. And so, in mammals, particularly terrestrial mammals, there's a couple different ways that it gets into us. First off, through a digestive tract. And as we take in extraorganismal fluid into the digestive tract, most of that's going to be pulled into the vascular fluid through the small and large intestines. So that's where most of that uptake is taking place. But then also some minimal amount will be taken up through the skin as well. This is actually a relatively unimportant method of taking up fluids, but this will happen as well. And for the most part, we are going to ignore this as we talk about this, at least in terms of mammals, when we talk about osmoregulation here. Then we also have a lot of different ways that we are getting rid of fluids all the time. So the major one, I'm going to list these from the most major to the most minor, and the most major one of these is the excretory systems of animals, is a major way in which we push fluids out of the body of an animal. And in most mammals, this is going to be the kidneys. All vertebrates, this is essentially the kidneys, and there's a lot of other animals that have essentially kidneys as well. So this is the major way in which we're excluding fluid from our body. Then we also have fluid that is going to exit us through the GI tract, and most of this is going to happen through feces. But also, if we have some sort of disorder like that you're vomiting or something else, that's another way that we can get fluid excluded from our body through the GI tract. Predominantly, under normal circumstances, feces is the way that happens. Also, the respiratory system. So as you breathe in, you breathe out, you moisten that air, and this is another way that you're losing fluids continually. And this tends to be just water, but there's a few other things happening there as well. And for humans, this happens while you're talking as well, not just respiring, but it's happening through the respiratory system. So as I'm sitting here talking to you about osmoregulation, I am also losing some fluids as well. We also have exocrine glands, so glands that essentially produce some secretion and then push it outside of the body. The major ways this is going to happen is through sweat. Also sebaceous glands, which are essentially your oil glands that are going to produce some secretions and push them out as well. Now, unlike your respiratory system, these exocrine glands are not only going to push out water, but they're also going to be pushing out some solutes as well. And so this is another way that we can be modifying the osmolarity or the osmotic balance of an organism as well. And we also have skin evaporation outside of the exocrine glands. So in addition to just like for instance sweat, where we're purposely pushing out water and fluids onto the exterior of skin for evaporation, there's other evaporation that just happens through the skin as well. And finally, there's a very minor amount lost through the reproductive tract as well. So there's reproductive fluids that are produced by organisms, and this is a very minor but also another way that we're excluding fluids from the body as well. Okay, so the major one that we talked about just now of outputs is the excretory system. And this is the one that we're going to be talking about for the next little bit, or for the majority of osmoregulation. And the reason is because this is probably the single most important system when we start talking about osmoregulation in essentially all terrestrial animals and the majority of aquatic animals as well. And so really osmoregulation is going to rely on our ability, or an animal's ability, to selectively excrete certain solutes in water. We need to get rid of the precise things that we want to get rid of and hold on to the things, and reciprocally hold on to the things that we want to hold on to. We can take in water and fluids and solutes in bulk kind of indiscriminately, not totally indiscriminately, but somewhat indiscriminately, and then rely on excretory systems to get rid of the things that are surplus or that we do not want. And that is the main strategy in which most animals maintain osmotic balance, have somewhat indiscriminate uptake, but selective excretion. And so we have several different sites of excretion. First off, especially in aquatic organisms, there's a lot of respiratory organs that are going to be doing some excretion. And so, for instance, gills, other things like this. This is a very convenient way for aquatic organisms to push water out of their body and also push solutes out of their body through respiratory organs. Also, we have some sort of excretion that's happening through the digestive system. We will have some things like in humans, biliverdin and bilirubin break down products of hemoglobin that are going to be pushed into the digestive system from the blood and be excreted as well. So this is another way, not a very major way, but a minor way in which we're getting rid of some solutes as well. Again, especially in terrestrial animals, the skin is a major way, is both in terrestrial and aquatic organisms. The skin is a major way, but in terrestrial animals, this is happening predominantly through sweat. We can sweat off water, but then also particular solutes that we need to dump off. In aquatic organisms, this is actually a fairly major way in which that they're going to be exchanging solutes with the environment. It's right there in contact with the environment, and as long as you can highly vascularize that, put a lot of capillaries into the skin, you can be fairly easily doing a lot of exchange with the environment through the water, and that works very well. However, the one that we're going to be mainly talking about here is a lot of organisms, even aquatic ones, have these dedicated excretory organs. In humans and other mammals, these are kidneys, or in fact, essentially all vertebrates, these are kidneys. But then we also have some other ones in other organisms that we'll talk very briefly about, probably especially in class, but maybe a little bit here in this the online lecture as well. So the purpose of excretion, when we talk about what we need to do with excretion kind of from the global level, we really want to maintain, number one, maintain the internal solute concentrations of solutes that are important to us. And the big ones that are important to life are going to be sodium, that should be a small a there, sorry about that, sodium, potassium, hydrogen concentrations, or pH again, pH is a solute here, and then also carbon dioxide, because again, carbon dioxide is going to be important when we talk about pH again. So these are really important solutes. This is not the only ones that we're maintaining or trying to regulate, but these are the major ones. And so when we start talking about excretory systems, this is going to provide a bulk of what we're talking about. Also, we want to maintain plasma volume. We need to maintain enough blood to be circulating through our body in order to not have too high blood pressure, but also not have too low blood pressure, and effectively move this blood through the body. If the plasma volume gets too low, the cellular component is going to stay about the same. Blood gets more viscous, and it's hard to push around, and so that can cause problems. Also, if the plasma volume gets too high, we have a limited amount of volume in our vascular system. And so if the plasma volume gets too high, you'll start having elevated blood pressure problems. And that can actually lead to a whole range of problems all throughout the body as well. So we need to maintain appropriate plasma volume. So that's another important purpose of excretion. We also want to be removing harmful substances. For instance, here, any metabolic waste that we've accumulated we need to get rid of. Also, any ingested toxins, other things like that. And then here's the big one, and this really is a kind of, the last or a subset of this, is we need to maintain osmotic balance. So this is kind of how we've been talking the entire time, how this plays into osmotic balance, but this is really kind of the overarching theme of excretion, is maintaining appropriate osmotic balance, but then also in particular substances as well. And to maintain pH. Like I was saying before, this is kind of a subset of maintaining internal solute concentrations, but really we can talk about this on its own as well as being kind of an independent phenomenon. It can go back and forth. And so, one other thing that I need to just draw your attention to as we're talking about all of this and how this can be adjusted is that nitrogenous wastes are going to present this special challenge when we're talking about excretion and talking about osmotic balance, because they're pretty toxic, to tell you the truth. And so there's going to be special consideration paid to how we deal with these as we talk about excretion. So we might as well just talk about now, so it's kind of setting the stage as we talk about this more later on. And so all catabolism, every time you break down nutrient molecules, we're going to be producing carbon dioxide. However, when we undergo protein catabolism, we have this added sort of byproducts that we don't have with carbohydrate catabolism and fatty acid catabolism, is that we're also producing ammonia. And ammonia is really, really noxious. It does a lot of bad things inside living organisms. It's been used for years and years and years as a disinfectant. I don't know if any of you are old enough to remember. It used to be a really common thing for mopping people's floors because of the disinfectant. I remember when I was young, my mom had ammonia that she would use to mop the floor to disinfect it. It's nasty smelling. It's just not good for life, and that's the reason that got used for that. One of the big reasons it does this is a really strong base. It disrupts pH pretty effectively, and pH is something that we really need to tightly control in life. And so we don't want a lot of this showing up inside our body. And it does disrupt pH because it is a strong base, and it will readily accept another proton. And when it does that, we become an ammonium ion. And so this is NH4+, and ammonium itself does a whole lot of bad stuff inside you. For instance, one thing it does is it'll interrupt sodium-potassium ATPases by interacting with the same area that the potassium interacts with, and clog up that entire thing. Now, the sodium-potassium ATPase is this really vital thing, as I'm sure you have caught on to by this point in the quarter, if not this point in your biological career. This is used all over the place. It's used in your neurons, it's used in your muscles, it's used in your nephrons, as you will see later. It's just really important. So anything that's going to be interrupting this particular transport, that's bad. Also, aside from this, ammonia is also a pretty potent neurotoxin. And so, in other ways, it's also going to disrupt the normal function of neurons. And so, overall, ammonium and ammonia, just not good. But this is a consequence, a buildup of this is a consequence of protein catabolism. And so we need to deal with it because it's going to be building up. Now, aquatic animals, this is actually not too big of a problem because they can just dilute this down. As they get ammonia and ammonium built up in their system, they can dilute it with more water, exclude this from their bodies, and everything's A-OK for them. So this actually is less of a problem for aquatic animals than it is for terrestrial animals. Now, terrestrial animals can convert ammonia to urea or uric acid through the urea cycle, or a special shunt of the urea cycle that gives you uric acid. I'm not going to spend a lot of time on that. If you're really interested in that, I believe Dr. Craig goes over that in biochem, so I'm not going to recap that here. But really, this is the way that terrestrial animals can deal with this, and this makes it into a less toxic form, but urea and uric acid still are not good. Urea does a really good job of denaturing proteins, and so we still do not want this around, and so we still need to get rid of that. So, at that point, that kind of sets the stage for excretion as an overall process. So excretion itself is then kind of broken down into 4 different processes that are going to happen. And we're going to talk about those and talk about some of the overarching themes in excretion and begin talking about the anatomy that goes into that. And that should probably be good for this lecture. So, let's talk about these processes. The very first process in excretion is going to be filtration. Filtration is we're going to take blood plasma and filter it out into the excretory organ, whatever that happens to be. And usually this is going to be without whatever cells are in the blood, and also without proteins. Just like when we were talking about ultrafiltration that's happening in capillaries, is very similar to what's happening here, although slightly modified, in that we're going to be pushing out most of this plasma with essentially all of its solutes, and the bigger components are going to stick in. But other than that, pushing essentially everything out. From this point on, to the point that ends up being collected in the urinary bladder, we're going to call it filtrate at this point. Because really it's just the filtered blood plasma. It's not urine yet. We haven't gone through the entire process of forming urine, and it's not really blood plasma anymore. So we call it filtrate. It's just kind of this intermediate filtered stuff. The next thing that's going to happen is we are going to undergo secretion, and this and the next process are really happening in tandem, but we're going to take other solutes that we don't want and move them into the filtrate. So if we have something else that didn't get filtered, or didn't get filtered enough, we're going to move those into the filtrate and build them up there. And so this is a way that we can put special things that wouldn't otherwise get filtered into that filtrate, or wouldn't get in there enough. Here's the other big thing, though. We also have resorption. And so resorption is going to happen as we take, or reabsorption. Excuse me, reabsorption. Reabsorption is happening as solutes are going to be taken up out of the filtrate back into the plasma. So a lot of these things that get filtered out, we want to keep more in our blood than just the cells and the large proteins. There's a lot of sodium in there. There's a lot of potassium in there. There's a lot of these other solutes that we really want to hang on to. There's glucose in there we want to take back up. There's all these things that we want to keep. And not least of these is... I'll come back and talk about that in a minute. But there's all these different things that we want to take back up that we don't want to get rid of. And so this is reabsorption. But then we also have osmoconcentration. Not only do we want to take these solutes back up, and we want to keep those, we really want to hang on to that water too. And so what we have is, through a countercurrent exchange mechanism, similar to what we were seeing earlier in like bird respiration, we are going to be able to concentrate the filtrate and take out as much water as we possibly can, because we want to retain that. Water is good. Water is good for life. And terrestrial organisms actually don't really have necessarily this huge surplus like aquatic organisms do, that we can just kind of toss away water willy-nilly. So we need to take as much of that back up as we possibly can. And our kidneys are really good at doing this. Nephrons are really good at doing this. And so what this means is that filtrate, through this countercurrent exchange mechanism that we have going on, will make the filtrate hyperosmotic compared to essentially all the other fluid components inside the body. So it's going to be hyperosmotic to the intracellular fluid, the extracellular fluid, and your vascular fluid as well. So we're pretty good. All terrestrial animals are pretty good at hanging on to as much water as they possibly can out of that. Now this has some consequences for maybe not everyday life, but life. You may have seen this person before. This is Bear Grylls, and he has had this documentary, kind of a reality documentary, I guess, where he is going through various survival situations. And he's gotten somewhat of a reputation as demonstrating the drinking of one's own urine during survival situations. And I am here to tell you, don't do that. This is a bad idea. Give your kidneys more credit than that. This one is just like drinking seawater. It is hyperosmotic to all your blood fluids, or all of your intracellular fluids. You put this in your digestive tract, and it is going to be drawing water, reversing the process of osmosis from going from your digestive tract into the vascular system, but the other way. And you will actually end up losing more water than if you had never drinking your urine before. It's just going to end up dumping out the other end of your digestive tract. And taking more water with it than you put in. This is a net loss in water. Not good. Bear Grylls gets away with it because at no point during that show is he actually on the edge of real dehydration. Is my guess, at least, why he gets away with it. But he also has cameramen following him around, so who knows? They might be slipping into water as well. But the point here being, don't drink your urine. Do not do that, please. Thank you. Okay, so now that we've gotten that out of the way, let's talk about some overarching themes in excretion. And this is really kind of like some ideas that are just going to tie together conceptually what's happening. I want you to keep these in mind as we go through the entire rest of the process of excretion and know generally kind of the ideas that are behind what's happening. So the first one is that really what we're doing is we're filtering everything out of the blood, except for the cells and proteins, of course. But we're filtering everything out, and then we're pulling back in what we want. Now, this is a really important concept, because what this means is that we don't need a special transporter for every random waste that happens to end up in your blood. If you eat something that gives you some crazy, something in your blood that shouldn't be there, we don't necessarily have to have some special transport that's going to pull that out. You'll pee it out. If you live that long, you'll pee it out, because through filtration it's going to come out, and unless there's something specific to pull it back in, it's not going to be pulled back in. And so in this way, we kind of have a system that we don't need to have specialized ways to detoxify absolutely everything that you could ever come in contact with. We have kind of like this, we're getting rid of it all and just holding on to a few things that happen to be important. Also, one of the big things that are going to be resorbed is sodium. And sodium is going to be really, really important that we're pulling out, because sodium is going to be the major osmolyte by which we undergo osmoconcentration later of water. And so sodium plays a central role in excretion by its reabsorption. So it's actually actively transported back out of the filtrate. And this is happening by a plethora of different transporters. A lot of different transporters. Not least among them is the sodium, potassium, ATPase. But then also we have some sodium, potassium chloride symporters. We have a whole list of things that are pulling sodium back out. And we actually do a really good job, even though there's still some sodium left in your urine. We can't get it all. Really, if you look at the amount that we get out, the percentage that we get is very, very impressive. And so we're taking most of that back up actively, and it's playing a really important role in osmoconcentration later on. And this is where water comes in. Water is going to be mostly passively removed, and this is going to be through osmosis. And again, our primary osmolyte that's causing that is going to be sodium. And we're doing this through a countercurrent exchange mechanism. So again, like we were talking about earlier, is that countercurrent exchange mechanisms make it so that you can really get the most out of passive processes like diffusion and osmosis. And we can do that because we can maintain gradients down to very low levels because of this countercurrent exchange mechanism. So this is going to be really important as we're talking about what's happening later on. Okay, so to begin off, we're just going to begin talking about the actual anatomy of what's happening here, and move on to more of that a little bit later. But really what's happening is that the nephron is going to be the basic unit of excretion. Later on, these are going to be making up of a kidney, but we'll talk about that later. And the principle of the operation of the nephron really isn't that different between most animals. So the basic operation here is going to be pretty universal in animals, although the anatomy is going to kind of change up depending on what group of animals that you're talking about. Sometimes we're talking about these things called flame cells. They're essentially small nephrons. They're individual. And instead of using our process of filtration, they're using these small cilia that drive filtration. Hence, these cilia beating give them the name flame cells. We also have protonephridia that are in things like a lot of mollusks. The basic thing that's happening here is very similar to what's happening in the nephron. Just the anatomy is varying there. And then also in nephron, what we have in essentially all vertebrates. And the biggest difference between the functionality of excretion is going to happen between aquatic animals and terrestrial. And the biggest point of this is going to be that there's this greater need for water sparing in terrestrial animals. So there's going to be a much tighter regulated excretory process happening in terrestrial animals. It's going to be very similar in concept in aquatic animals, but we're going to have this big lack of this great need for water sparing in aquatic animals. So that's going to be the big difference there. And altogether, nephrons are going to occur in vertebrates in kidneys, and these specialized excretory organs we call kidneys. And these kidneys are going to, in humans, contain about in the neighborhood of 1 million nephrons, about 0.9 to 1.1 nephrons for normal human beings. And so that's a lot. We have a lot of these very small functional units that are coming together to make this kind of total functioning organ. And the other thing is that there are going to be no new nephrons formed after birth. And so whatever nephrons you have in your kidneys at the time that you were born are all that you're going to have throughout your life. And so really this isn't a tissue that's going to be turned over in terms of these functional units at all. Okay, so let's talk a little bit about the actual anatomy of a nephron. And we can divide it initially up into two different components. We have this here, this is the vascular component. And then we also have this part up here, which is the tubular component. Now, the vascular component is going to have this area, this arterial coming in. It's going to go into this little rat's nest of capillary beds, this tuft of capillaries, and an arterial going out. And this is the afferent arterial coming in. And then we have this structure here, which is the glomerulus. And then we have this arterial that's heading away from this tuft of capillaries of the glomerulus, which is known as the efferent arterial. Now, if we're looking at an actual picture, what we would see is that the afferent arterial is a lot larger than the efferent because a lot of the volume of the blood has ended up in the tubular component after it leaves here. But then what we have is after the efferent arterial leaves the glomerulus, it's going to come and wrap around the remainder of the tubular component of the nephron. And I'm showing this wrapping around a little bit. In truth, this forms a very dense net around the entirety, not just the little part here. It's going to form this really tight net around the entire nephron for the rest of the length, including up here and over here. This is just a simplified diagram. In fact, I should let you know, this entire thing is an overly simplified diagram. Nephrons do not actually look like this. They are much more complicated, much more convoluted than what you're seeing here. So we have this capillary bed that's going to be all around the rest of the nephron. This is what's known as the peritubular capillaries. And these are going to be heavily involved in the reabsorption and the secretion of stuff back into this tubular component. Okay, so let's talk about the tubular component here. It really comes in two different sections here. We have this part here that includes the glomerulus, but then also the part around this here, which is called the Bowman's capsule. But this is, I'll come back to that in just a second. This is the renal corpuscle. But then we have this other section here, all this tubing that's leading out of the renal corpuscle, and all of this is what's known as the renal tubule. The renal corpuscle, the renal tubule. So really the renal corpuscle is made up of the glomerulus, but then also this part of the nephron, the tubular portion of the nephron that's around that, which is known as the Bowman's capsule. And the tubular component is really divvied up into three different parts. This initial part here, which is what is known as the proximal convoluted tubule. After that, we have this thing that dips down and comes back up. This is what is known as the loop of Henle. And then we have this third section here. So the loop of Henle comes from here down up and then approximately stops here. And we get this last section here, which is known as the distal convoluted tubule. Okay, so we'll come back and we'll start talking about this more in the next lecture, but I'm going to go ahead and leave it off there. And yeah, we'll come back to this more later.